Josep Carreras, Research Institute.
Okay, can I start? Okay. Good morning, everyone. Well, as he said, I'm Gerard Godoy, and today I'm going to talk you about how vitamin C triggers NF-kappa-B-driven epigenetic remodeling on dendritic cells. So first, I will introduce you a cell that do not need to be introduced, but I will do it anyway. Dendritic cells are a specific immune cell that have the ability that after recognition of a PAM or a DAM, present the antigen to the T cells and starts the immune response, the adaptive immune response. And as you probably know, they can be divided in several subtypes that they are mainly differentiated in the bone marrow. But in our lab, as we are more epigenetic lab, we are more interested in this specific subtype that are dendritic cells that came from monocytes. So there's well-known monocyte-derived dendritic cells that differentiate when the monocytes arrive to the tissue. What makes monocytes interesting, and this is why we choose this model, is that they are highly plastic. So depending on the cytokines that they are in contact with, they can differentiate to several cell types. For example, not only dendritic cells, also Langerhans cells, macrophage, heterogenic dendritic cells, or even osteoclasts. This plasticity, it's mainly given by epigenetics, but I will introduce you today one of the most known epigenetic mark, which is DNA methylation. And this epigenetic mark consists in the addition of a methyl group in the cytosine in a region of the genome called CpG island. And the way of how this epigenetic mark works is that when this mark, so this methyl, is present in a promoter region of a gene, the expression of this gene is blocked. And to modify this epigenetic mark, there are two main enzymes that are the NMT and TET enzymes. The first one, the NMT, is the one adding this methyl group to the cytosine, and then TET2 is the one removing it by oxidating this methyl, and then by active demethylation, this methyl group is removed. As an enzyme, this enzyme uses, for example, iron as a cofactor, and we know that vitamin C oxidates iron, and so then if we oxidate the iron, this iron can be used by TET2, so by vitamin C, we are increasing the activity of this enzyme. So knowing that epigenetic regulates monocyte to dendritic cell differentiation, and vitamin C is able to increase TET2 activity, as we have previously described in our lab, we asked what will happen if we add this vitamin C during this differentiation model. So what will happen if we increase the activity of this enzyme? So let's move to the results. So when we did the DNA methylation analysis of this model, so here you have monocytes, dendritic cells without vitamin C, and here with vitamin C, so in orange, like the oranges. So during this differentiation, there are already a demethylation, but when we add the vitamin C, this demethylation is highly decreased, not only during differentiation, which is this first M1 cluster, also during the activation, that is the M2 cluster. What was kind of shocking for us is that all these changes that the vitamin C was producing were also include the changes already produced during the normal differentiation. So we were just increasing something that's already happening in the cell. Then when we did the gene ontology analysis from these two clusters, we observed in the first in both clusters, categories related with immune, with dendritic cell function. For example, I don't see if the quality is quite good, but anyway, so for example, leukocyte activation, dendritic cell migration, etc. Then, as I said, we specifically saw how TET2, one of the three variants of this enzyme, is the one playing a role during this differentiation. The problem with TET2 is that do not have DNA binding domain, so this enzyme needs to be recruited by another transcription factor to make their function. So to study which transcription factor could be recruiting TET2, we perform a Homer analysis that consists in to study the transcription factors that bind to the regions where we observe a DNA methylation change. And at the first cluster, we observed EGR2, which is the already described transcription factor regulating the differentiation of the dendritic cells. And in the cluster 2, we observed NF-kappa-B or P65, that is also already described transcription factor related with the activation of the dendritic cells. Okay, so this is for at the DNA methylation level. So then we move to the expression level. So we perform an RNA-seq with the same conditions. At what we saw, we could differentiate all these genes in 6 different clusters. And as you can see, in all the clusters except for one, the addition of vitamin C is producing highly increase in the expression of all these genes. And when we perform gene ontology analysis with all this cluster of genes, we again observe categories related with dendritic cells, dendritic cell migration, T cells activation, etc. So we are like just increasing the function of the dendritic cells by the addition of this vitamin C. But then we ask, okay, we know that this is improving, but which of the DNA methylation changes is really affecting the expression of the genes? So to analyze that, we correlate each CpG with their closest genes, and we studied the expression of these genes from these 2 clusters. And as you can see again, there is a slightly increase in the expression of these genes, but this increase is more strong during the activation process. And also, when we studied enrichment of the methylation genes at the level of expression, we observe how cluster 1 are enriched in the cluster A1 and 2 of expression, and cluster 5, cluster M2 was enriched at the cluster 5 of expression. Then when we perform a Godoy-Tena analysis, that is a similar analysis as in the Homer, where we study which transcription factor could be regulating these clusters, we observe again at the cluster 5, P65, that it's the same one that we observed at the Homer analysis and the cluster 2. So we decided to focus on this transcription factor because it could be the one regulating TET2 and make their function. So to study that, we studied the expression of P65 during the activation, not only without vitamin C, also with vitamin C. And also, as I said before, well, they need to interact with P65, so we perform a co-immunoprecipitation of P65, and we observe how P65 was really interacting with TET2. Okay, so, well, you probably know how this is really affecting the dendritic cells. Okay, so to analyze that, we perform an in vitro study where we wanted to study not only the T cell activation capacity of the dendritic cells, but also their antigen presentation capacity. And to study that, we obtain PBMCs from a healthy donor, and we expand the T cells under several rounds of activation in contact with a specific antigen. In this case, we use the SARS-CoV-2 antigen. And after several rounds of activation, we obtain the clones, and from the same donor, to avoid the TCR cross-reactivity, we obtain the monocytes, and we differentiate them to dendritic cells, and after activate them and loaded with the correct antigen, we perform the co-culture, and we study the proliferation of the T cells. And when we did that, as you can see, well, we stain the T cells with CCCA, so you know that when there is proliferation, the levels of this marker is decreased. So as you can see, when we didn't add the antigen, there was no proliferation, but when we add the antigen, there was proliferation. But when the dendritic cell was differentiated in presence of vitamin C, this proliferation was increased. As you can see here, as a main result of several experiments, where the proliferation of the T cells when the dendritic cells were in contact with the antigen and also with vitamin C is almost the positive contrast, that there were T cells differentiated, activated with anti-CD3 and anti-CD8 bits. Then also you're maybe wondering where this knowledge can be applied. So I want to present you this therapy that is called dendritic cell vaccine for cancer immunotherapy, where it consists to obtain monocytes or premature cells from a donor or from a patient and differentiate them to dendritic cells, and then load these dendritic cells again to the patient together with the tumor-associated antigen. So maybe if we add vitamin C during the differentiation, this therapy could be improved. And so right now we are starting a collaboration with a group from New Jersey to try this in an acute myeloid leukemia mice model, to see if this is really affecting if this is really improving the dendritic cells vaccine therapy. Well, I wanted to say that this paper was published just 1 month ago, so if you want to have more questions, there's the paper. And I want to thank members of my group, and especially Octavio, that was the one who starts this project, and thanks you for your attention.
There are some questions? No? What? No? Thanks.